louder because here in front we cannot hear people in the back. And can we ask the lecturers to repeat the question?
Yes.
Oh,
sounds good. All right. Morning. So, okay. So where we left off last time, we were talking about associating polymers or associative polymers. And so just a quick recap where we were. The green light is on. How about now? Ooh. Okay. All right. Right. So our picture is that we have some polymers. They have some binding sites. And we said that these binding sites are mutually exclusive, so they can only make one contact at a time. And this is perhaps a more realistic model of certain types of biopolymers, such as nucleic acids that can hybridize, or very specific interactions, for example, between RNA and RNA binding proteins, where there's really a one-to-one interaction where we have the associations between these polymers. And so what we were doing was trying to write down the excess chemical potential for a system like this using the technique described last time. Okay, and so our expression looked something like this. All right, so there's some probability of overlap, and we're going to ignore this because we say that we can only have one monomer at a time on a lattice site. And then if we do not have an overlap, then we have to deal with this question of are the associating binding sites actually forming bonds, right? And so for this, we said that we have a total of F times L binding sites per polymer. F is the fraction of monomers that have binding sites, L is the length of the polymer. And then we can either have a randomly inserted monomer oriented in such a way that it happens to be facing a binding site on an existing binding site in the solution. So that gets some energy delta U, or that doesn't happen. And there's no change in the energy. Okay. So that's where we were. And we said that so far, we haven't really made any significant approximations, but within a mean field context, we can say that this probability of finding an adjacent available binding site is a simple expression if we assume that all of the positions and orientations of the binding sites are uncorrelated. If we do that, we get the probability that an adjacent lattice site has a monomer with a binding site times the probability that that binding site is oriented in the right direction times the probability that the binding site is not already occupied. It's not already engaging in a one-to-one interaction with something else, right? So that's this X here is the fraction of unbound binding sites which is a new variable that we're going to have to solve self-consistently for. But if we put this all together, we're going to get a rather simple expression. So this is our expression for excess chemical potential, assuming that we have this mean field assumption saying that positions and orientations of the binding sites are uncorrelated. Okay. And so this looks like what we saw before with the Flory model. This is just due to the fact that we can't put monomers on top of each other. But then we had this new term here, and this is the associative part. Okay, so what we have to do is deal with this fraction of unbound binding sites. We need to solve self-consistently for that quantity at equilibrium. And the easiest thing to do here is just to use some intuition from chemistry and how you describe a reaction between two monomers and a dimer. Right? So if we assume that everything that all the binding sites are uncorrelated in terms of positions and orientations, then we just have effectively an ideal gas of binding sites. And so let's use this sort of notation where we say we have two binding sites, let's call them A, can reversibly make a dimer, which is AA, right? So if we think about the way we would normally treat this in chemistry, we would write this ratio for a reaction quotient should be equal to an equilibrium constant. And this equilibrium constant here is going to be something that is related to the partition functions of the dimer and the monomers, right? So these are just, if you had just an independent isolated dimer, it has some partition function, it's going to have some energy, right? That accounts for this, the fact that there's a bond being formed here, right? And then this would be the partition function for just the monomers. And so what this should end up looking like, I'll explain where this comes from. This ratio should be precisely this quantity. This is a symmetry factor. Because when you have this dimer, you can rotate it, right? And it's the same in both directions. So we have a factor of 1/2 in this partition function, right? We can only form a dimer if the two things are oriented in the correct direction, which gives us this 1/z factor, right? And then this is just the Boltzmann factor that accounts for the fact that we only form a bond if we have if we have our two binding sites touching each other, right? So this looks like the expression for second virial coefficient. This is exactly how you can calculate a dissociation constant between two molecules using statistical mechanics. And so this is precisely the expression that we want for describing this. All right. Okay, so yeah. Oh yeah. Yeah, our chemistry. Chemistry here means ratio of concentrations, right? Just to make it look like something that commonly appears in textbooks. V is the volume, right? Yes, from last time. V is the volume of a lattice site. We need this for dimensional reasons, right? Because we have a concentration divided by concentration squared, we should end up with something that has units of volume, and that's here.
Chemistry, they write the ratio of the two, they use detailed balance and write a ratio of the two rates.
Right. So this also can be related to the ratio of on and off rates, which gives you an equilibrium constant, which is again precisely this, right? So kinetic interpretation, here's your equilibrium stat mech interpretation, just in terms of ratio of two partition functions, right? Okay, so I think that the key thing to realize here is that if we're going to assume that we're making a mean field approximation here, that everything is uncorrelated, then we should be thinking of just about dimerization of an ideal gas. So this is the thing we're looking for, which necessarily requires this. Yes, please. Where the -1 comes from? Fantastic question. So the -1 is necessary because when we write down, I guess what we're implying here by this delta U is this is the energy change when you form a bond, right? We're saying that if you put two binding sites facing each other, then you absolutely have to form a bond, right? So if they're oriented in the right direction but not forming a bond, that would be because the energy is infinite, if that makes sense, right? So we necessarily have to subtract off 1 here for this to be consistent. In other words, look at the equilibrium constant that we have here in this expression, right? If you were to put in delta U equals 0, and you didn't have the -1, then this would be a non-zero expression, which would say that you have some fraction of dimers. So the -1 ensures that if you say that a bond has 0 energy, means that the bond can't form, then it says that I have 0 concentration of dimers. There are a number of different ways to argue this. Another way is when you write out the statistical mechanics expression for a dissociation constant or a second Vero coefficient, you also always have a -1. This is this Mayer F function, which again, it accounts for this same thing. If you wish, you could write this as e to the minus beta times an energy, where you define the energy such that you equate these two expressions. And that would mean that the bonds do not form when that new energy you've defined is infinite, right? The delta U is very much defined. We're just trying to be consistent between what we mean here and here, right? So up here, so the delta U is the change in the system energy when we form a bond, right? But I'm by writing out this expression up here, I'm saying I can only form a bond if the two binding sites are facing each other, right? So in that case, my equilibrium constant expression must have this -1 in it in order to say that the only way to form a bond is by having a negative delta U as opposed to just a finite delta U. No, this is just your energy of binding. Okay. Yes.
It's not obvious to me why that would mean none of them are forming bonds, because wouldn't that mean that it's just as cheap to be in a bonded pair as it is to not be in a bonded pair? Like there's there's no energetic benefit to forming the bond, but there's also no penalty.
Absolutely. But then you have to be clear when we say here about when we made this distinction between having an adjacent bonding site and a binding site that's not adjacent. Do we have, we're saying that we necessarily form a bond if your binding site is adjacent to another binding site. So we're not saying that the delta U has to have any particular value. We're just simply being consistent between what we mean there and what we say here. Okay. So if we do that, then we can just write a conservation expression. Conservation of binding sites. We know that the total volume fraction of the system where we have binding sites must be related to these concentrations that we wrote down for different species. Okay. We have a factor of 2 here because there are 2 binding sites in any one of these dimers, right? And so if we just plug in the expression that we had over there, we get this line, which comes from our expression of the chemical equilibrium of binding sites. And the key thing is that this X here is now appearing because this X is defined to be the fraction of binding sites that are available, right? So those are the ones that correspond to binding sites that are not in these dimer pairs. Okay, so now hopefully you can see where we're going with this. If we simplify this expression, we can divide through to end up with this. And if we take all of this factor here and define this association parameter, then we can write this. So this is now just an equivalent way of writing the chemical equilibrium expression that we had here, but now in terms of our X variable, right? The one that tells us about binding sites being available. Okay. And with this in hand, we can finally simplify our expression for the excess chemical potential, which was here. You'll see that this reduces to this is our key expression for associating polymer theory. It says that we have the initial contribution from not putting monomers on top of each other, but everything to do with forming bonds simply goes like the total number of monomers that have binding sites on any given polymer times the logarithm of the available binding site fraction. Okay. Okay. So let's look at what the phase behavior of this model looks like and how this can be different from what we had with the Flory model, and under what circumstances you would actually observe these differences. Okay, so the first thing we should do is solve the chemical equilibrium equation, right? So in this case, chemical equilibrium equation says we have this quadratic equation, right? And so if we plot the solution, everything is dependent on this one parameter, F times phi times delta. In this direction, we have very few very weak interactions. Here we have very many very strong interactions. Right? And so this ends up decaying something like this. In this regime here, we have approximately something that is decreasing linearly. We call this the weak binding regime, right? Because if we randomly choose any binding site, chances are it is not going to be forming any associative interaction. Right? Whereas if we come down here, then we have a different asymptotic behavior. And this is our strong binding regime. Okay. So we should look at what the phase behavior of this model looks like in these two different regimes. It turns out that in this limit, this weak binding limit, everything looks exactly like the Flory-Huggins model, even though we've made this assumption that we can only have one-to-one binding. So to see that, we should simply plug this expression here, this expansion for low f theta, f phi delta, into this part here, right? So we can call this just focusing on this dissociative contribution. Here we have this is the weak regime. We have this. And so if this parameter is small, we can Taylor expand, and we get this. So the thing circled is our association parameter times the fraction of monomers that have binding sites squared. That is going to play the role up to a factor of 2 of the chi parameter that we had in the Flory-Huggins model. So the prediction is that even though you have a system where we have, in principle, a heteropolymer, we have some binding sites, some monomers have binding sites, other ones don't. We have directional one-to-one interactions. As long as you have relatively weak interactions, your phase behavior should be indistinguishable from that of a system where you just had isotropic pairwise interactions. Okay, so you can ask, when is this valid? And clearly you need to, according to the Flory model, you need to have a big enough effective chi parameter in order to be above the critical point and thus have phase separation. But at the same time, we can't violate the assumption that this F phi delta is small. Otherwise, we wouldn't be in this regime where we can use this expansion, right? And so if you put those two things together, you realize that this only works if you have phi over F much less than 1, right? Which simply says that I need to have very long polymers with very many binding sites in order for this to work, right? So the correspondence between these two models only works in this long polymer limit when you have lots of different binding sites, because then you can still have phase separation with any individual binding site having a low probability of actually forming any sort of associative bond. Okay. So more exciting things happen if we focus on the strong binding regime. Okay, so in this case, we want to take our asymptotic expression here and stick it back into our excess chemical potential. And if we do that, we get that we do this, we see that that this associative term ends up looking like this, right? Which it's a bit more informative if we think in terms of the osmotic pressure, right? So we can use the Gibbs-Duhem relation again. Our associative contribution to the pressure in dimensionless form is, we have to do this integral right? So if you recall, this is this standard thermodynamic relation between the pressure and the chemical potential at equilibrium. And if we just solve that here, we get, again, an extremely simple expression. Okay, so this is the contribution to the osmotic pressure if you're in this strong binding regime. The V? This is times the volume, just to make things dimensionless, times the volume, yeah. Yeah, okay. Right, okay, so what's interesting about this? There's no delta, right? There's no association parameter anymore. We say that once the bonds become strong enough, your thermodynamics here doesn't depend on how strong those bonds are because of this assumption that you have one-to-one binding, right? Everything that can be associated is associated, and this is going to change your phase diagram, right? In particular, it says that if you keep going to stronger and stronger bonds, you are not going to change the you are not going to change the properties of your condensed phase, right? You've basically formed all the bonds you can make, and so if we make a little plot, so there's going to be some critical value of our association parameter. If the association parameter is stronger than that, we can have phase separation. But what is interesting here is that this tells us that we're going to have a condensed phase volume fraction that becomes a constant as we make this interaction very, very strong. And that constant can be less than 1. So you can end up with a system that looks in the condensed phase like this. Lots of open space with the polymer taking up potentially a very, very low volume fraction. And cranking up the interaction strength even more doesn't change this picture. So that's what we see here. Condensed phase. And this is our coexistence region between our two phases. Yes. Yeah, fluctuations. Great. So fluctuations in the sense that we should go beyond mean field. Is that? Yeah. Right. So fantastic. And of course, there were a lot of questions last time too about when can we use these mean field arguments. In general, this idea that things are uncorrelated only works in condensed phases. So here, we should ask, how condensed is this, right? Our accounting of these one-to-one interactions takes care of the fact that the binding sites can either be bound or not bound, right? But this argument that the excess chemical potential has uncorrelated positions and orientations of monomers will break down if you say have very non-mean field like interactions in the regions between these binding sites, right? And so in particular, if you have chains here that are in the swollen chain regime, they want to expand and push these binding sites far apart. Under those conditions, then the sort of mean field picture can break down. Right? So that's one possibility. Another possibility is that you can form intramolecular bonds, and I will come back to that in just a second. Because there's one more thing I'd like to say about this plot before we move on. And that is that so far we've been focused on phase separation, meaning this equal chemical potentials, equal pressures between our two phases. But there's something else that can happen here too. It's not the same as phase separation, but rather just making a gigantic component of a system spanning network, right? So it's possible, this picture that I drew here, to get this to get a picture that looks like this, even without phase separation, meaning your entire system looks like this. So we want to estimate when this happens. This is called physical gelation. So the idea is that as you increase the concentration of your polymer, you should expect, very much an analogy to what Marcus talked about yesterday, you should have initially a dilute solution with mostly free polymers, then maybe you'll get some clusters, then at some point you will have a system spanning network that forms bonds like this, regardless of whether you actually have phase separation to form spatially inhomogeneous droplets. So we want to estimate when that happens. Right, so the basic idea is that if I'm going to have a polymer that's part of some system spanning cluster, then it has to engage in at least two contacts, right? One contact it has to have in order to be part of the cluster in the first phase. And then the second contact would be to recruit something else into the cluster. And if you have more than two contacts, then you can get a system spanning cluster. So when does this happen? It's the point at which the probability of some binding site in your gel, which is just 1 minus this fraction of bonded interfaces here. This must be equal to this at this gel point. Okay, so again, this thing here is the total number of binding sites on any given polymer. And we're subtracting off 1 because in order for one polymer to already be part of a cluster, it already has to be using at least one of those bonds. So this is saying that on average, our polymer is going to be making at least one more contact with its remaining binding sites. Right. This is something that is determined only from this binding site chemical equilibrium. And if you just plug in what we had over there, and do the algebra and get this expression. So we get another curve on this plot, which is our gel line. Okay. So this is what's known as a sol phase. Is your gel. So what this says is that 2 different types of things can happen in a system with associating polymers. Let's consider what happens if you're below this critical binding strength, right? You don't form droplets. You don't have different regions of the system with different polymer concentrations. We're below a critical point, so the polymer concentration is the same everywhere. However, if your concentration is below this pink dash line, then you will have everywhere in the system individual polymers or small polymer clusters. You go above this point, and you have a system spanning network. So your entire system looks like this. Right? If you then move up into the phase separated region, what it's now saying is that everything over here is going to look like this picture inside of our droplet. So now we have a system where we have, say, a droplet inside of a lower concentration phase. But in here, we have this system spanning network. And out here, our low density phase or low concentration phase is still a sol. Okay. So this is the physical picture predicted by this simple mean field model. Yeah. Yes, exactly. And yeah, so percolation in the sense that we go from having, if you want to follow connections between bonds, they don't span the system, and then at this point, they suddenly do. But this is not a true thermodynamic phase transition. In this model, none of there are no discontinuities, right? So nothing is truly a infinitely sharp transition. Again, very much an analogy to what Marcus was talking about yesterday. And the other key thing here is that if we're below this critical point, the whole system looks the same. Anywhere you look will look like a gel or like a sol, right? It's only once you go above this point that what you see depends on where you look, whether you're looking inside of this droplet or outside. Yes. That's a great question. I left that as a homework question. I think physical intuition should tell you, but yes, indeed, for at least for this model, we have explicit expressions for both, so you can check. I don't want to give it away. All right. So there's one more possibility that I'd like to discuss briefly. Okay. So continuing on with this theme of when can we have, when is this sort of mean field uncorrelated treatment valid? We should also consider the fact that we can form intramolecular bonds, right? And this becomes extremely important in a dilute phase, because in the dilute phase, we pay a lot of entropic penalty in order to bring polymers together. Once we've done that, it makes sense that they would form as many bonds as they possibly can, especially if we're in this strong binding limit, right? So the picture here is that if we are in the dilute regime, we should probably expect the system to look much more like this. Between, say, small dimers, or maybe something that's folded like this, right, where it can truly form intramolecular bonds, as opposed to having a system spanning network, because this costs a lot more entropically to bring many more polymer chains into close proximity. Okay. So this idea here immediately violates our mean field assumption, right? To treat this, you can no longer say that our binding sites are, that their positions are uncorrelated in space, because we're saying precisely the opposite, right? If I find one associative bond here, I'm actually quite likely to find other ones nearby. So this gets complicated, but there's one quite simple thing that we can do, and that is to treat different species as different species, right? And establish chemical equilibrium among them, right? So if we particularly focus on the possibility of folding, right? Then we can say that in a dilute phase, we can have chemical equilibrium between this unfolded polymer U and a folded polymer that looks like this, right? And then we just want to generalize what we've done for this excess chemical potential. So it now depends on whether we have a folded state or an unfolded state, right? So we can write this, right? So we have two different species now, folded and unfolded. This part here, again, our not allowing things to overlap. When you try to insert a new polymer chain, it doesn't matter whether the things you potentially overlap with are in the folded state or unfolded state. So we just are 1 minus the sum of these volume fractions here, right? But it's only in the unfolded state that our binding sites are available to engage in further interactions. Here, no such binding sites are available because we assume that everything is folded, right? This is a model that should work reasonably well if we have very strong interactions, right? So we're going up in this direction into this strong binding regime. This sort of picture is what you would probably expect in the dilute phase. So we need one more expression because we now have introduced a new species. So we need to have overall chemical equilibrium between the two. Right? This is our. In fact, there's only actually one type of polymer, so we have to have chemical equilibrium between the unfolded and folded states. The key thing is that our ideal contribution to the chemical potential now depends on whether the individual molecule has no bonds or many bonds. So what we should do is say, strong binding limit, only 2 species, right? Of course, it's an approximation, but this should work pretty well if we go to very strong interactions, right? So what we should have is, we now have to account for these terms. Right? These concentration independent terms in our ideal chemical potential, right? The internal energy of this thing is different from this thing. In fact, it has FL divided by 2 bonds here, right? So we should have some contribution here. And so what we should expect is that as you increase the bonding strength, you will shift your equilibrium from this state to this state if you're in a dilute phase. Okay. So what's going to happen? One more piece of chalk. Again, this state here is going to tend to compete with this one, but it's only this one that can phase separate, right? So the fact that you can form this intramolecular folding should oppose phase separation, and it should act to stabilize your dilute phase, right? So if we come back up here, what's going to happen is that our dilute arm here at some point may turn around and come back in this direction, right? Because as I go, if I'm down here, I have mostly unfolded state. Everything is just the same as before. I can basically ignore this chemical equilibrium associated with intramolecular folding, right? But as I make this association parameter stronger and stronger, I should expect that in the dilute phase, I'm going to overcome this conformational entropic cost to fold, to be able to form all these intramolecular bonds. And so I should get this F state, right? And ultimately, as I go in this direction, I'll have the same number of bonds in approximately in both the dilute phase and the condensed phase, right? In the condensed phase, I have this system spanning network here. But in the dilute phase, I have an equal number of bonds because I can do this. Okay. Right. So in this regime, the key thing is that you can see a competition between the formation of these small structures, say folded structures or small oligomers that are in your dilute phase that oppose phase separation, tend to prevent the formation of these system spanning networks because these bonds can be formed intramolecularly at a much lower entropic cost. If you go all the way in this direction, you'll eventually have roughly the same number of bonds formed in both phases, and it really comes down to the details of the conformational entropy involved in the polymer, which goes beyond these sort of simple arguments we're making here. But overall, you can get a dilute phase boundary that turns around, which means that you can get reentrant phase transitions. So if I sit here at some polymer concentration, I initially start in a sol phase. I make my bond stronger and stronger. At some point, it phase separates into a sol low density phase and a droplet, that is my high density gel phase. And then if I keep going, eventually I end up with a single phase system again that is just these folded polymers in dilute solution. Okay. So a hand, yes. Depletion effect from, right. Yes, so in this simple argument here, we have neglected the difference in these excluded volume effects due to these different species. We've just added them up, which is again consistent with this overall mean field argument, but should be refined indeed. Yeah, and it's not too difficult to do so. Yeah. Yeah, this qualitative picture, if you have sufficiently strong either folding or formation of these self-limiting small clusters, then this sort of reentrant behavior is going to be observed regardless of those effects. Yes. Yes. Well, these ones have more, the monomers have more translational entropy, right? But the gel can have more conformational entropy because there are more ways that these polymers can arrange themselves to form bonds, right? And so that's why the precise picture depends on these details, depending on solvation effects of these sort of spacer regions, among other things, right? But that's why this entropic accounting becomes quite tricky and essential. Okay. All right, so can I plug this in? This one?
Oh, I guess I can do this one. No, I got it.
All right. Any further questions on what we have so far? Okay, so what I'd like to do is just show two examples of well-studied systems that occur in living cells. We've really been able to map out these phase diagrams and show some of the features that we've explored with these mean field models. Okay, so what is this system? This is a very cool engineered phase separating system that is controllable by shining light onto these cells. And the key thing to remember is that all of this is taking place in living cells. You can see that here the outline is essentially the outline of the cytoplasm. Right. Okay, so two key proteins have been introduced and greatly overexpressed. One is a protein that forms essentially an inert core, right? This core is an oligomer, right? So it's actually composed of 24 monomeric subunits. And what happens is that when you shine a light on the system, blue light, those monomers self-assemble to form a 20-former. Right? In addition, there's a second protein that has been attached one one protein per monomer, so that when you form this oligomer, you end up with a little hairy ball, right? And these proteins sticking off of this core are, in this case, intrinsically disordered proteins that can undergo phase separation in the absence of being stuck onto this core. But here, the fact that this engineered correlate system exists allows us to reversibly create and dissolve these droplets. So the reason this was initially introduced about 7 years ago was to map out phase diagrams inside of cells. And so we can see a lot of the same features that that we've been discussing, right? Okay, so right. So on this axis here, we have this core to IDR ratio. All right, so IDR, these are these sticky proteins, these polymers that don't form any stable structure. The core is this 20-former, right? So what this axis is telling you as you move down is the number of, on average, the number of these sticky hair-like particles that are stuck onto a core. And so the F parameter is precisely the same thing we've been discussing, except now F can be greater than 1 because we can have up to 24 of these sticky binding sites on any particular monomer. Okay, so in this picture, this delta parameter, this association, is just set by biology, right? The interactions between these various proteins. So the two things that can be controlled experimentally are the concentrations of these two different molecules, the core forming monomers and the sticky IDRs. So as we move down, we're going into this more phase separating regime, right? So we had this effective chi parameter that went like F squared delta, right? So as we move down here, we're going to higher F squared delta. We're going farther into this phase separating regime. Okay. So the different points here from the experiments show where phase separation is or is not observed. The filled circles meaning that phase separation is observed, empty meaning it's not observed, and some estimates of what the coexisting polymer concentrations are, core concentrations in our two different phases. And you can see that as you approach roughly this point where you've saturated the entire surface of this core with 24 IDRs, you follow this picture that looks qualitatively like what we have for this weak binding association regime. So you can actually fit that in this way. Right? So this is a really nice demonstration, and actually being able to measure phase diagrams in cells that are currently alive is very cool. Down here. So at this point, the system becomes more complicated than this, or our description should become more complicated than what we've had so far with only one macromolecule plus solvent, because now we have an excess of these sticky IDRs, right? So you really should be thinking about a system with more components, right? So we have now solvent plus 3 IDRs plus these sticky cores, right? And so these sticky IDRs are going to compete with these fully formed cores to reduce the concentration of the cores that are in this condensed phase. So essentially goes beyond, we can effectively think of this as a single macromolecule and solvent picture up here, and that breaks down here.
very elongated.
Yes.
So are these, can this be thought of as fluids or more solid like behavior?
So it's both, and it depends on the length, the length and time scales that you're probing, right? So these things do flow. The best way of convincing us of that is just to look, right? So you can see that this is sped up, but not tremendously so. These things are clearly flowing and fusing on the time scale of minutes, and this looks very much like liquids, right? Again, the time scales are long compared to what you'd have for, say, oil and water, and that's in large part here because the surface tensions between these different biopolymeric phases are much, much lower than you'd have for hydrocarbons and water. But if you probe things on much shorter timescales, comparable to the, sort of the lifetimes of these intermolecular bonds, then things can look much more elastic, right? And so, indeed, and people have explored this with passive and active rheology and really characterized the fact that there is a crossover here. In this case, of course, they're in cells, so it's hard to say exactly. You can do this exact same thing in vitro, and they do in this paper, and you still see the same like rough fluidity. This is most certainly reversible because at the end of this video, which is on a loop, you see that the light is turned off and it all dissolves. Yeah. Interestingly, it goes, you see the color changes first because the core and the IDRs are tagged separately. So there's a disassembly followed by a dissolution. Okay. So for one more example, we can look at something that's a bit closer to actual biology. From this really nice paper a couple years ago, investigating the sequence dependence of the phase behavior of an intrinsically disordered protein, A1-LCD. So LCD means low complexity domain. It's low complexity because even though you have an alphabet of 20 amino acids, actually only a few amino acids are highly overrepresented in the sequence here. Right? And so many of these proteins in biology that don't fold into compact states but stay disordered are LCDs. This one's of interest because it is a prion-like domain. Right? So it is something that can induce the folding of other proteins. Right. So the key thing to note here is that first off, this in vitro measurement of the phase behavior can be fit extraordinarily well by this simple Flory-Huggins model. And so this is precisely what we've seen, this black line here, two different types of experimental measurements, or sorry, three different types of experimental measurements for these points. And at first glance, this might be a bit surprising because our Flory model assumed homopolymers, right? But this is clearly a heteropolymeric sequence. The key idea is that if we are in this weak binding regime, then everything gets averaged out. These mean field approximations of everything being roughly uncorrelated hold up quite well. And you really can fit this effective chi parameter, effective Flory chi parameter to describe this. And further, if you start to change the sequence composition, you can modulate the phase behavior, right? So here we have this the amino acids that are highlighted in yellow are the ones that are stickiest. These are very hydrophobic amino acids. They don't like to be near water and they tend to drive phase separation. So what they show here is that, again, in vitro, if you change the overall sequence composition, then you can use this Flory-Huggins model to predict the shift in the critical temperature and these phase boundaries as a function of sequence. Okay. All right. The other thing you should notice here is that if you look at the relative spacing of these very hydrophobic amino acids, they're not spaced randomly. They're somewhat evenly spaced one apart from each other. And so if you ask the question, what happens if we group these amino acids into different spatial patterning? Does this change the phase behavior? Okay, right, so here we have this, the wild type, which is fairly close to what they call perfect spacing, right? Completely uniform spacing. Sort of the other extreme would be having this very blocky sequence where you have clusters of these sticky hydrophobic residues. Okay, so what should we expect based on the associating polymer model? Well, in this case, if we think about this patchy sequence, we should have, if you think about renormalizing this, we effectively have a small number of these binding sites that collectively engage in much, much stronger interactions and are interspersed with these relatively inert linker sequences, right? So this should be much more like the picture shown over here, right? But we should then worry about whether this sort of mean field picture from this Florey-based analysis actually works, or if we're going to see something that looks much more like this gel transition, right? And indeed, that's what happens. You end up in a region of your phase diagram where, if you consider this patchy sequence, you no longer have fluid droplets. Then you see that they're not spherical anymore, which indicates that you don't have a significant surface tension trying to form these droplets. Instead, it looks much more like a system-spanning network of these essentially these cross-linked polymers, right? So this is a nice demonstration of what happens when you move from this weak binding regime to this strong binding regime just by scrambling the sequence. Okay. All right. So any other questions on that? Okay. So I'd like to take the remainder of the time today to talk about multi-component systems, right? So far, everything has been only talking about a single macromolecule in solvent. But that, while we can explain some of the features of these experimentally observed phase diagrams, it's not possible to explain this exquisite organization that you can get through the formation of these membraneless biomolecular condensates. Right, so for that, we absolutely have to move to a mean field picture, sorry, multi-component picture. And so that's what we'll do next. So the first thing I'd like to do is focus on this important question of what's in and what's out. What is actually going into these various condensates and what's not. Experimentally, you can measure these partition coefficients, right? So this partition coefficient is defined as the ratio of the concentration of a protein or nucleic acid inside of one of these structures, I guess this one, to everything outside. And what you generically see is that you have a large number of distinct gene products that selectively partition into various condensates. Okay, so how do we relate this partition coefficient to things that we've seen? Well, for our multi-component system, this partition coefficient for some molecular species I is simply defined to be the ratio of the concentrations or number densities inside and outside. Okay. But we also know that in general, in order to have phase coexistence, we need to have equal chemical potentials. That has to be established for each molecular species individually. So therefore, we can write that the chemical potentials are the same inside and out. We can use our decomposition into ideal and excess chemical potentials. Like this, right? But recall that mu ID is just KT log rho I. And again, we can forget about the concentration independent term because whatever we have on this side is going to cancel what we have on this side. Okay. Right, so we can just rearrange this equation and we get that we move the ideal parts onto the same side, we get this. Where this thing here is precisely our partition coefficient, right? Okay, so what you are looking at when you see the data shown here. Yeah, absolutely. What it means, yes. So we have two phases that we're going to focus on for the moment. Of course, there are many present, but we're focusing on one particular phase and what it's in direct contact with, which was the rest of the cytoplasm, right? So this is in, that's out, right? What we're measuring here is the ratio of the concentrations in versus out, right? So any number greater than 1 means it's partitioning in. These numbers can be gigantic, meaning that you have a much, much higher concentration of a particular say protein or RNA. And what this shows is that that number is just directly related to the difference in excess chemical potential, right? So in these multi-component systems, this quantity here is really the key thing of interest because it tells us where these molecules want to go, right? Yes. It's out when it's in. Thanks. Okay. All right. So the other thing that we can do is think about what phase separation might look like in systems of multiple different types of polymers that can all undergo potentially, phase separating with one another due to their various interactions. So the simplest thing we can possibly write down here is a multi-component version of our Flory-Huggins model. right? And so in this case, the chemical potential of any particular species is this. Okay, so to get this expression, go back to what we had yesterday, and note that there are only two minor changes. One is that when you try to insert a polymer, again, it doesn't matter what is taking up space. So the phi that we had before is replaced by the sum of all of the volume fractions of all the possible polymeric species. And now our interactions, instead of being an energy times the volume fraction of a particular polymer, is just the sum over all the polymers that can be in this species, right? So again, everything here is again precisely the same mean field arguments. Right? And so you can use this to interpret this partitioning data to try to extract what these various interactions are, these epsilon ij's, right? And so I'm going to write it this way, as a note. Here, we're using the dilute solution as a reference state. Which just simply means that the interaction of solvent with itself or a polymer with solvent is taken to be zero by definition, right? So we say we have an isolated polymer and solvent, and we call that the zero energy. Then we only need to sum here over in polymer species. There's no solvent in this, just the polymer species. Where does this come from? This came from yesterday. So it's a generalization of what we had before. If you, oops. No, it's a mistake. It's a typo. Thank you. Yes. Okay. Sorry about that. Right. Okay. Sorry about that. Okay. Very good. So we have, yes. So we have this one being our excluded volume, and this one being our mean field interactions. Okay. Okay. So in order to have phase coexistence in this model, we could potentially have more than just two phases. We still need to establish the same coexistence conditions. Right? So we have equal temperatures, equal pressures in all of our different phases, and equal chemical potentials for all components in all phases. So the alpha is the phase index, right? And the I some molecular species. Okay. So all of these things must hold for all phases and all molecules. But recall that there's this other important condition, which is that we have to have stability. And so that means that we have to have second derivative of our free energy must be positive definite. Right? Or equivalently, we can write right? We must have everywhere in our system, we must have this matrix has no negative eigenvalues, right? This is what's required in order for our system to obey the second one, right? And so if we use our free energy, our Flory-Huggins free energy, this just becomes so we can actually learn a lot about what types of phase diagrams we could potentially get by thinking about what the possible eigenvalues are of matrices. And the simplest possible case we can consider is when we have two different polymer species. So we have two polymers in solvent. We can try to draw out what these phase diagrams might look like. So we have two polymers. Oh yeah. Can I put it up in just a second? Can you see it? This one. Yeah. This one goes up. Yeah. Very good. Genius. All right, thank you. All right, so we have two polymers in solvent. All right, so we have four possible types of phase diagrams. That we can reason based on those types of eigenvalues. Okay, so this is a phase diagram at constant temperature, or probably better for us here, this is constant average energy, right? We're not changing the average energy in each one of these. We're just varying the volume fractions of our different polymer species. So the simplest thing we could have would be an interaction matrix where all of our polymers, all right, so interaction matrix is proportional to just all minus one, right? So this says that all of the polymers interact the same way with each other. Everything should should look the same regardless of whether we're looking at phi1 or phi2. This phase diagram thus will look something like this. We'll have our coexistence region. We have a dilute region here and a condensed region here. Right? So if you move in this direction, increasing our different polymer volume fractions the same together, you can just treat this exactly as a single polymer solution because all the interactions are precisely the same. If you project this onto this axis here, you just get a phase diagram that looks exactly like what we had before. Okay. Simplest possibility. More interesting possibility would be something like this. So this means that our polymer species 2 attracts itself, right? It wants to phase separate, but the other polymer species effectively acts the same as the solvent, right? So we should get phase separation in this direction, but not in this direction, right? So this ends up looking like something like this. Coexistence region here between a phase that's dilute in phi1 and phi2, and here condensed in phi2, right? So this would be a case where the partition coefficient of phi2 of species 2 is really high here, but species 1 does not want to partition into that phase. And again, you can get a sense of what's going to happen here by just reasoning from this interaction matrix. I have one strong direction, and all the other ones don't want to do anything. Right, so the third case we can consider would be one where the polymers don't care so much about the solvent, but they really hate each other. So if we did that, we could get some repulsive interactions off diagonal, something like this. This gives us a phase diagram. Looks something like this, right? So the instability is going this direction now. The two polymers want to get away from each other. So if we put enough polymer into the system, moving up in this direction, then we will get demixing the two different types of two different condensed phases. This condensed phase is mostly polymer type 2, and this condensed phase is mostly polymer type 1. Okay, there's one more possibility. And that would occur if we choose something looks a bit like this. So this means that polymer 1 likes itself. It wants to aggregate, right? Or it wants to form a condensed phase, and so does polymer 2, right? So just by taking this picture here and flipping it, I might try to draw something like this. Right? Okay. But that's not what's actually going to happen. Right? In all of these cases, we have a coexistence region where we just have two different phases, say overall dilute condensed, or condensed mostly of polymer 2, or 2 condensed with different types of polymers. But here, we're going to get 3 different phases. So this one wants to go like this. We have one instability in this direction, another instability in this direction. Okay, so this whole thing here is your coexistence region, and we have 3 phases. So our picture, we're going to have our system. We have, if we put ourselves somewhere in the middle of this coexistence region, we will phase separate into a dilute phase, and then one condensed phase here, and some other condensed phase here. Right, where the 2 different condensed phases have different polymer compositions and different partition coefficients for these various species. Right? And so the upshot is that by controlling these sorts of interaction matrices, even within this relatively simple mean field picture, you can start to see how you can get a wide variety of distinct phases. You can control their different partition coefficients by tweaking these interactions here. And this is really our emerging understanding of how you can get compositional control and self-organization without membranes, basically by playing with this picture, but with many, many more dimensions than just 2. Yeah. Which orientations? The best way to do this, to get some insight, is to think about this matrix here, which is linear in epsilon ij. And think about what its unstable eigenvalues would be. So unstable here would be the ones that are less than 0, which would violate our stability condition. And that would tell us where the system wants to separate. In order to just draw these things a bit more easily, just using some intuition about what likes to be with what and what doesn't like to be with what, right? So a negative number here means going to a lower energy, right? It's stable to have things near each other, right? So this one by symmetry has to look like this because there's no difference as I go around this axis. Here, the picture is simply that only polymer 2 is going to want to phase separate. The other ones are 0. And we said the convention is that 0 means you're the same as the solvent, right? So the phase separation has to go in the direction roughly parallel to phi 2. That's the only obvious possibility. This one here is saying that the polymers don't want to be near each other. They're repelling each other. They don't really care so much about themselves. You could, right? And so if you do that, you get some picture like this. But if you make the polymers attract each other, but not want to be together, you make this more extreme by putting +1s here, right? Then you have now 2 different types of instabilities, right? Because it's just this thing twice, but now rotated, one instability going up, one instability going over this way, right? This point here would separate into these 3 points, right? And the amount of polymer within each of these phases is given by these points, right? And the overall amount of phase alpha, phase beta is given by mass conservation where you are within this triangle. Yes.
So if you're at a different point, not the point is like the intersection.
Pink point's an arbitrary point. I could have put it anywhere. Anywhere inside here, right? So this is the generalization of, before when we said we're in the coexistence region, you split into 2 phases. Now the coexistence region is actually a higher dimensional space within this phi space. And so therefore, again, it doesn't matter where you are inside, you will phase separate at equilibrium into these points.
So like if you're, for example, like if you go to the second picture and you change by 1, like slightly, right? You're going to phase separate into 2 slightly different points.
That's right.
But there you always get the same point.
Also true. Yes, yes, right. So here, yeah, just to repeat that, here, there are only two phases, but I have multiple degrees of freedom. So if I had an overall system volume fraction 1 and 2 that's here, then I would end up with two different points on this binodal, but which two exactly would be the ones that satisfy both mass conservation and my equal p's and mu's, right? So I think easy to understand in practice, so easy to understand in principle, in practice, lots of equations to solve, right? But here, because we have three different coexisting phases, you no longer have any additional degrees of freedom. Once you say that I'm inside this triangle, I know that I'm going to have these 3 points. It's only a question of how much of each phase I'm going to have. Yeah. How do I know I only have 4? Because in this system where I only have these two possible sets of instabilities, right? They're just qualitatively speaking, there are only so many possible pairs of eigendirections. Yeah. Yes.
Matrix of this quadratic Li of J, that's responsible for your
I multiplied everything by Lj because I have put the delta ij over rho I instead of multiply through by Lj just to make it look a bit more symmetric. Otherwise, you'd wonder what's special about I. It's different from J.
What is the situation if different columns below each other?
Yeah, it should look a bit more like this. No, you're right. It should be somewhere in between. Yeah, somewhere between this one and this one. So perhaps we should say 5.
Then within each one, can you have this phase separate either into polymer 1 or species 1 or species 2, but then each one can phase separate concentration. Sort of dilute.
Mhm. Right. So in order to get this, in order to be up here, I have to have an overall very high volume fraction of polymers, right? In order to be in this coexistence region at all. And so at that point, if this is my picture, I have my two different phases, which each one technically still has solvent polymer 1 and polymer 2. It's just that this one is mostly polymer 1, this one's mostly polymer 2, and there's a little bit of solvent in both. But there's very little solvent because I have an incompressible system and I'm already at very high polymer volume fraction.
Get one phase coexistence dilute that feature.
Yes. Yeah.
Simple.
Yeah. I think, obviously there are, yeah, you can come up with more detailed more scenarios, right? If you wish to go into more and more distinct cases, generally speaking, I think it's a reasonable way of thinking about how you can get to multi-phases. But in this particular case, this is really the polymer melt case. So if we go all the way to this point here of having no solvent left, we would only just have polymer 1 and polymer 2. Yes. They're non-reciprocal. Okay, so our matrix here has to be has to be symmetric, right? Because we're talking about the interaction between I and J doesn't matter how we phrase it, Ji or Ij. This truly is the symmetry is truly a constraint in these systems, right? Like if you go back to where this came from, counting the number of contacts between these different monomer types that are placed together, we're just counting the number of contacts. So we really cannot have an asymmetry around this axis. Where are the tie lines? So all the tie lines should go like this. The key rationale being that if you that it shouldn't matter how you project this into any linear combination of phi1 and phi2, because energetically speaking, 1 and 2 are the same. So it must have this sort of look. Good. Let's break. All right. Thank you.